Assalamualaikum. The topic which we'll discuss today is fungi and agaricus. What are fungi? Fungi derived from Latin word fungo mean flourish, used with reference to mushrooms. About 5,100 genera and 50,000 species of fungi known today. Eukaryotic meaning that they have membrane-bound nuclei and organelles. They are spore-bearing, heterotrophic organisms that produce extracellular enzymes and absorb their nutrition. Fungi body plan are unicellular, filamentous, or both mean dimorphic. Hypha is the basic cellular unit in filamentous fungi. They may be septate or syncytic aseptate, collectively a mycelium. Limited tissue differentiation and division of labor, somatic and reproductive structures. Placentenchyma, all organized fungal tissue, somatic and reproductive. Nuclear status of fungi. They are eukaryotic, uni, bi, or multinucleate, haploid, diploid. Diploid nucleus is less frequent. Monokaryon means one nucleus per hyphal compartment. Dikaryon means two nuclei per hyphal compartment. Homokaryotic, heterokaryotic. Organelles. Plasmalemma. Unit membrane, protein layers separated by lipid layer. Mitochondria. Endoplasmic reticulum, Golgi body is composed of 4 or 5 flattened cisternae. Cytoplasmic microtubules, cytoplasmic movement, maintenance of cell shape, microtubules. Vacuole, young hyphal-like vacuole bounded by tonoplast, ribosomes, synthesis of proteins. We can also see the diagram of mushrooms in which the structured cap, scale, green ring, stalk, volva are formed. Hyphae, cell wall, well-defined chitin lacks cellulose, fungus chitin, polysaccharide, glucosamine, pectin protein, lipid cellulose, scalose, minerals also present. Nutrition. Heterotrophic secrete extracellular enzymes, absorptive nutrition, saprobes, decayed dead organic matter, pathogens, biotroph, nectotroph. Some points are parasites, commensals, and mutualists. Spores are the minute propagative unit function as a seed but differing from in that a spore does not contain a preformed embryo. Fruiting body, any complex fungal structure that contain or bear spores are sporocarp. Reproduction in fungi. Sexual reproduction occurs when hyphae from one fungus fuse with hyphae from a fungus of the opposite mating type. The fused hyphae then form a reproductive structure such as the mushroom. Inside the structure, nuclei from the two mating types fuse. These diploid nuclei undergo meiosis and produce haploid spores that are released. Asexual reproduction. Asexual reproduction occurs when specialized hyphae produce haploid spores by mitosis. The fungi that develop from these spores are genetically identical to the parent. Fungi that do not have an observed sexual stage are grouped together and called imperfect fungi. Exogenous and endogenous spores involved in the asexual reproduction of fungi. Fungi life cycle. Mycelium in fungi life cycle. Mycelium divides in mitosis to produce spores and they germinate again to produce mycelium. This occurs in asexual reproduction. And in sexual reproduction, there are three main phases: plasmogamy, karyogamy, and germination. In plasmogamy, haploid cells from two different mycelia fuse to form heterokaryotic cell with two or more nuclei. You can also see the eukaryotic states. Then karyogamy occurs, in which the nucleus fuse to form a diploid zygote. Zygote then forms meiosis and produce haploid spores. Haploid spores again germination to produce a multicellular mycelium is formed. Reproduction. Vegetative reproduction. The most common method is vegetative reproduction. The hypha breaks up into small fragments accidentally or otherwise. Each fragment develops into a new individual. Reproduced vegetatively by other means such as fission, budding, sclerata, rhizomorphism, etc. In fission, the cell constricts in the center and divides into two, giving rise to two individual. Budding is commonly found in Saccharomyces. These buds arise from the protoplasm of the parent cell and ultimately become new individuals. Economic importance of fungi. Yeast is used in brewing beer, edible mushrooms, harmful fungi, rhizopus, bread mold, spoils bread and other food. Athlete foods. Fungal infection of the skin between the toes, potato blight can destroy an entire potato field. Effect of fungi on crop production. The fungi play both a negative and positive role in agriculture. Damping of disease. The seedlings of almost every type of plant grown as a commercial crop such as tomatoes, cotton, mustard, peas, beans, tobacco, spinach, etc. are prone to this disease. It is caused by a species of python. The potato blight. Late blight of platters is another destructive crop disease. It does a great damage to the potato tubers. Downy mildews of grapes, apple scab, red rot of disease of sugarcane. Eager disease of rye. It is an important disease of cereal crop rye. It results in the formation of poisonous celosia in the rye kernel. Brown rot of stone fruits. It causes enormous losses in the fruit crop of apricots, cherries, plums, and peaches. Rust diseases. They attack our cereal crops and forest timber. Some of them, such as black stem rust, yellow rust, and orange rust, are a serious threat to our wheat crop. Poor fungi. They are the common wood rotters. They destroy timber and lumber. These are the negative effects of fungi on crop production. Now we can study the positive effect of fungi on crop production. They also bring about rot, decay, and decomposition of animal and plants remain, releasing plant nutrients in form of available to green plants as food. The soil fungi utilize many inorganic salts. These are prevented from being lost from the soil by leaching. Some fungi form mycorrhizal association with the roots of certain plants and help them in their nutrition. Such plants will grow satisfactorily only when the mycelium of the appropriate fungal partner is present in the soil. The fertile soil contains twice as much living fungus cell material as the material from the bacteria and other soil microorganisms. Gibberellin produced by Gibberella fungiculae is used as growth hormone, accelerating plant growth. Aflatoxins, the most potent carcinogenic agent, are produced by Aspergillus fumigatus, parasitus, and Penicillium islandicum over dried foods and groundnut meat. Effect of fungi on industrial applications. The industrial uses of fungi are many and varied. Alcoholic fermentation. These are brewing and baking. Both are dependent on the fact that the fermentation of sugar solutions by yeast produces ethyl alcohol and carbon dioxide. Enzyme preparation. Invertase is extract from Saccharomyces cerevisiae. It has many industrial uses. It hydrolyzes sucrose to a mixture of glucose and fructose. Preparation of organic acids. The important organic acids produced commercially as the result of biochemical activities of molds are oxalic acid, citric acid, glucanoic acid, gallic acid, and fumaric acid. Vitamins. A number of molds and yeasts are utilized in the synthesis of ergosterol, which contain vitamin D, riboflavin. Effect of fungi on industrial applications. Antibiotics. Certain fungi form an important basis of fermentation of coca beans. Gibberellin is used to accelerate growth of several horticultural crops. Cheese industry. Certain fungi, popularly known as cheese molds, play an important role in the refining of cheese. They give cheese a characteristic texture and flavor. Manufacture of proteins. The yeast, Saccharomyces cerevisiae, and Candida noodles contain high percentage of protein of great nutritive value. Preparation of organic acids. The important organic acids produced commercially as a result of biochemical activities of malts are oxalic acid, citric acid, glucanic acid, gallic acid, fumaric acid, etc. A good many fungi synthesize fat from carbohydrates. Now we can discuss the subtopic of fungi, which is agaricus. First, their taxonomic position. Kingdoms, fungi, division, Amestiogo, Mycotina, subdivision, Basidiomycotina, class, Holobasidium mycedia, order Agaricales, family Agaricaceae, genus Agaricus, species Apicomplex. Habitat. Agaricus species are commonly called meadow mushrooms since these are typically found in open, well-manured, grassy areas. The genus contains some of the fine edible mushrooms, including Agaricus campestris, Agaricus biporus. Agaricus campestris. Agaricus campestris, Latin campestris means relating to plant or flat filed. It is common edible and cosmopolitan species. It grows in damp and dead organic substances such as humps, horse dungs, damp rotten wooden logs, tree trunks, and meadows indicating organic matter. Plant body. The plant body of fungus can be divided into two main parts: the vegetative mycelium, basidiocop formed of hyphae sexual reproduction. You can also see the anatomy of mushroom agaricus, in which the cap, gill, stem are the main parts. Vegetative mycelium. The mycelium may be primary mycelium, secondary mycelium, tertiary mycelium. Primary mycelium. It is a monokaryotic, short-lived and develops from germination of basidiospore. The hyphae are septate and hilate. The cells are short and contain oil globules, vacuoles, and thin protoplasm. Secondary mycelium. It is a dikaryotic and evolved by dikaryoticization through smatogamy or two compatible monokaryotic hyphae. The hyphae composing secondary mycelium are long, branched, septate, and have short cells. Typically, doliopore septa are present between adjacent cells. The secondary mycelium is more abundant, perennial, and produces mushrooms year after year. Tertiary structure of mycelium. The mycelium produces a basidiocarp, and mushrooms are sometimes referred as tertiary mycelium. It forms complex tissues which produce basidia. The mycelium form microscopic, thick, tangled, woody masses called spores. Reproduction. The fungus reproduces sexually usually. The definite sex organs are absent. Plasmogamy is done by fusion of two compatible somatic hyphae which results in the formation of a dikaryon. The nuclei of the dikaryon to produce dikaryotic secondary mycelium, which we can also see in the picture. The basidiocarp development from the hyphae of subterranean perennial secondary mycelium. Basidiocarp. The aerial part of the fungus is called fruiting body. Basidiocarp of the fungus is popularly known as mushroom. Structure. Pilus. A mushroom is umbrella-shaped, generally the upper portion, called pilus of carp. It is circular and expanded. In young condition, the pilus is more or less button-shaped and is completely enclosed by a thin membrane called velum. Stipe. The pilus is brown on a fleshy pinkish white stalk called stipe. As the pilus grows in size, the velum ruptures and it remains round the stipe called annulus. Lamella or gills. A cavity is present under the pilus in which large number of thin vertical plate-like structures called lamella. Gills are the first pinkish, but as the spores mature, they become dark brown in color. Development of basidiocarp. It starts as a tiny swelling consisting of a knot of the subterranean hyphae of the secondary dikaryotic mycelium. This swelling enlarges into a broadly oval structure, basidiocarp. Then a ring-shaped cavity is formed, prelamellar chamber. The portion of the primordium above the prelaminar cavity finally develops into the pilus, and below portion is called stipe. Internal structure of basidiocarp. It is composed of interwoven hyphae which run parallel to each other in stipe. They are compactly arranged to form pseudoparenchymal tissues in the pilus region. In stipe, the central hyphae is loosely arranged, with prominent air spaces forming the medulla, and the outer hyphae forms cortex. The pilus also shows similar arrangement of hyphae into the central medulla and outer cortex. Some of the hyphae present in pilus development into the lamella. Structure of lamella. Each lamella consists of three parts: hymenium, trama, and subhymenium. Hymenium. It is the fertile region of lamella consists of club-shaped binucleate cells forming a palisade-like layer on both sides of lamella. All the cells of the hymenial layer are not fertile. Others are developed to form basilia and others are sterile. They develop to form paraphyses next to them and elongated cells. They protrude beyond the paraphyses and are called cytidia. Trama. Internal to hymenium is called trama. It consists of multinucleated elongated cells that extend from hyphae of the pilus. These hyphae are so orientated that their long axes lie parallel to the hymenium. Subhymenium. In between the hymenium and trama, there is region composed hyphae arranged at right angle to those of trama. The cells of these hyphae are isodiametric and binucleate. We can see the development of basidium and basidiopores and discharge of basidiopore by a small drop of liquid. This is also discussed in the previous slides. Fairy rings. The fairy rings, commonly formed by the field mushroom Agaricus campestris, often measure about 6 feet in diameter. Marasmus aureatus, which is commonly known as the fairy ring mushroom, forms very large but irregular rings. When the mycelium becomes mature at tips, sporophores are produced. These sporophores appear in a circle. These circles of mushrooms are commonly called fairy rings because of an old superstition that the mushroom growing in a ring indicates the path of dancing fairies. Life cycle of mushroom, which starts from the spores. Those spores germinate in the primary mycelium, which goes to form the secondary mycelium. Then secondary mycelium goes underground to produce a mushroom, which is called first button. Then the mushroom of Agaricus formed. Stipe gill cap. If we zoom the portion of gill, then we can see the basidium. In basidium, the pair of nuclei is present which fuse to form zygote. Diploid nucleus undergo meiosis to form basidium, in which basidiospores are also present. Lastly, there are some references which we can also study about fungi. Thank you.